Would you would you please play your slides so we know that there is no technical issue?
Sure. Can you see it?
Yeah,
Is the video playing smoothly?
yes. But um I think it's little bit, sort of wider for the screen. So if maybe you can zoom out a little bit.
I don't think I can zoom out. I'm not sure how to do that.
Is this the only video in your presentation, or are there any other videos as well?
There's a few. Can you read the text clearly here?
Yeah, absolutely.
Okay, great.
So I think we can go ahead with it rather than, if I
Sure.
mean, it's okay, it's no problem.
Okay.
Right. Bismillah ar-Rahman ar-Rahim and Assalamu alaikum and good day, ladies and gentlemen. I'm Hazima Mozam and I welcome you all to an exciting webinar organized by Comstech. Today, the speaker of our webinar is Professor Dr. Mustafa Mir. Professor Dr. Mir is associated with University of Pennsylvania, USA. Professor Mir received his PhD from electrical engineering in electrical engineering from University of Illinois in 2013, where during his graduate work, he invented new microscopes that provide high-resolution quantitative phase images of the living cells. He applied these new microscopes to a variety of problems in medicine and the basic life sciences, ranging from clinical pathology of the blood samples to single-cell growth and neural development. He did his postdoctoral work at University of California, Berkeley, where he studied gene regulation in Drosophila embryos using a combination of advanced microscopy methods, especially single molecule imaging, biochemistry, and genomics. Dr. Mir built a customized lattice light sheet microscope to capture the first data on the transcription factor dynamics within the nuclei of living and animal embryo. Dr. Mir started his lab at UPenn in January 2021. His lab focuses on developing and using advanced fluorescence microscopy methods and analysis and analysis tools to study gene regulation and nuclear organization during the early embryonic development and in disease. Dr. Mee received the National Institutes of Health Directors New Innovators Award in 2021, and he was also selected as an inaugural Freeman Hrabowski Scholar by Howard Hughes Medical Institute in 2023. The title of his talk today is Molecular Kinetics of Gene Regulation During Embryonic Development. With this, on behalf of Comstech and myself, I welcome you, Dr. Mir, and would again thank you for your time and for waking up too early in the morning for us and to deliver this lecture. Over to you, sir. Thank you.
My pleasure. Assalamu alaikum, everyone, and thank you for this opportunity to share my work with all of you today and to the organizers for putting this together. So I understand that this is an audience with probably diverse expertise, so I'll take some time as I go through the talk to provide the necessary background material. And I encourage you all to ask questions in the chat. I'll try to keep an eye on it as we go along, so we can address them to make sure everybody's understanding the material. So the goal of my lab is to build and apply new technologies to understand how the nuclei within living cells are organized and how this organization changes during embryonic development and how it might go wrong in disease. And I'll explain what I mean by this. And the end goal is that if we understand this in a quantitative and mechanistic fashion, we can then manipulate this nuclear organization for therapeutic and bioengineering applications. So what is the big challenge with nuclear organization and why is it so hard to understand? So as many of you likely know, all of the cells in our body have the same genomic information, which is encoded in our DNA. And if you take all of our human genome and you stretch it end to end, it's about 2 meters long. And this genome has to be then packaged and wound up and stuffed into a nucleus, which is on the order of 5 millionths of a meter in diameter. And this nucleus resides within a cell, which is then about 10 millionths of a meter in diameter or so. So there's an incredible compaction problem that happens here. Now, on top of this, the different cells in our body, of which they're on the order of 37 trillion, they all have to access the genome within their nuclei differentially. And depending on what type of cell they are, they have to read different genes that are encoded along the DNA to express different proteins. For example, your brain cells have to make the proteins necessary to make a neuron. Your skin cells have to make the necessary proteins to make skin. Red blood cells have to have the necessary proteins to carry oxygen. And all of this information, the information necessary to encode all of this function is within our genome. So the interest in our lab and many people in the field is understanding how this information is read out in a robust and reproducible manner to make these decisions. And in my lab, we focus on the first part of this problem, which is the initial reading of DNA to make messenger RNA, which is called transcription. This messenger RNA then goes on to make proteins, of course. So the challenge is transcribing the right genes in the right place so that you make all the different cell types that compose our bodies, for example, at the right time so that embryonic development proceeds with this beautiful clockwork-like choreography that we see in all animals, and at the right levels so that we avoid developmental defects, for example. And then how all of this comes together to make and maintain a living animal. Now, almost every single gene in humans is transcribed by an enzyme, a protein called RNA polymerase II. Now, on its own, this protein, Pol2, is rather promiscuous. So if you give it the right piece of DNA, it will transcribe it. And so a lot of the game in regulating transcriptional activity is about getting RNA Pol2 to the right place at the right time and regulating its levels of activity. Now, over the last several decades, we have revealed a complex and ever-expanding web of mechanisms which has evolved to regulate transcriptional activity. And each layer of complexity that is sort of displayed in this textbook-like diagram is representing decades of work in different labs. And really, a lot of hard work has gone into producing these types of models. And they've taught us about the importance of everything from how the DNA polymer is packaged into a fiber called chromatin, how within this chromatin fiber, there's spatial organization of the genome, and then on regions outside of the pieces of the genome that encode for proteins, there's non-coding regions that regulate gene activity and recruit regulatory proteins, which are called transcription factors, which is what we'll be talking about today. And these transcription factors bind to the genome and then recruit the other proteins necessary for the transcription reaction to take place. Now, the nucleus actually does not look like this, right? This is a textbook diagram. In reality, the nucleus looks more like this. It's a highly crowded environment in which the entirety of our genome and all the proteins that act on this genomic substrate are packaged into a relatively small volume. And so the challenge is understanding in this crowded environment, how one of these regulatory proteins, like a transcription factor, navigates this environment to find and bind a specific genomic target, and then occupies it with enough specificity to recruit the rest of the protein machinery that is necessary to carry out this biochemical reaction of transcription at the right gene, at the right place, at the right time, in the right cell to ensure that proper development proceeds. Now, over the last decade, it has also become clear that we need to move beyond in vitro studies of this process, like crushing up these components and running biochemical assays, to understand what are the mechanisms that exist to mediate this process in a robust manner. We need to study these dynamics within their native contexts at molecular scales. The challenge with this is that this entire scene that is being depicted over here is in around a 100 nanometer square, 100 billionths of a meter square. And so if you were able to label these proteins and then take a picture, this would occupy a single pixel on a typical high-resolution microscope setup. And because of the diffraction limit of light, which we'll discuss briefly, you would see just a gray square within a field of other gray squares, so making it impossible to access these molecular scale interactions that we are interested in. And then, of course, we don't want to study these in isolation. We want to put these in the wider context of a nuclear environment, which is constantly in flux, and where in the context of development within these nuclei, genes are constantly being read and self-fate decisions are being made as the embryo begins to take shape and then prepares for independent life. So how do we begin to tackle these problems and measure these kinetics in their native context experimentally? And this is really a story of building on decades of progress in really exciting technology development. So Sidney Brenner, who won the Nobel Prize in 2002, I think put it very elegantly, that progress in science depends on new techniques, new discoveries, and new ideas, and probably in that order. So I'll just go through some of the key technologies that enable our experiments. First of all, many of you are likely familiar with the use of fluorescent proteins that were originally discovered in all sorts of organisms, but in particular types of jellyfish that are bioluminescent and also express fluorescent protein molecules. And Roger Sheehan and others won the Nobel Prize in chemistry in 2008 for their discovery on how to extract the sequence for this gene and use it as a reporter in all sorts of contexts. For example, you can use it to look at protein expression in bacteria or in mice. And since then, people have isolated or developed a palette of fluorescent protein molecules that we can then use for different studies. So we can have many, many colors to look at different proteins. Now, these fluorescent protein labels now allow us to look in our images with molecular specificity. So we can attach different fluorescent protein labels to different proteins and then look at how they interact. And just very briefly, the way this works is that this molecule absorbs a photon that you can provide with a laser or a LED or some light source like that. The electrons get excited to a higher energy state. They lose some energy through non-radiative transitions, and then they drop down back to a lower energy state, and in that process emit another photon, which we can then detect as fluorescence. Now, to actually use these to study protein interactions, we use genome editing, which many of you have likely heard of, won the Nobel Prize in 2020, which is using the CRISPR-Cas9 system to genome edit basically anything we want nowadays. And what we do is we insert the sequence for the fluorescent protein we want to use next to the sequence of the protein that we want to study. These arrows depict the beginning of the gene. And when that gene is read and the protein is made, now you have a fluorescent protein attached to the thing that you want to study. So this way we can choose the fluorophore we want to use, and we can make sure that the protein that we are tagging is being expressed at the native levels within the species that we're interested in studying. And then we stick this genome edited embryo or cells under a microscope. And this, I'm going to very briefly introduce fluorescence microscopy here. The basic idea, as I mentioned, is you send your light source in, it bounces off a mirror, it goes into your objective of your microscope and excites fluorescent molecules within your sample. These fluorescent molecules absorb the photons and then they emit fluorescent light, which gets collected by the same objective lens. And then it goes through this mirror, which is a special mirror called a dichroic. So it lets the fluorescent light pass through, but does not let the laser light pass through. And the fluorescent light then goes and is collected onto your camera where you make an image. So this is the basics of fluorescence, which has been used for a long time now. In live cell imaging, the problem is that these molecules do not repeat this cycle forever. They undergo a process called photobleaching, in which after a certain number of these cycles of excitation and emission, you permanently lose the signal. And in this process, they also release free radicals, which are actually bad for the health and the viability of the sample. So there's a delicate balance on how often you want to image or how you image so that you can image for long periods of time and also avoid problems with the health of your sample. So this is why in my lab and in many labs now we are very focused on developing on applying new types of microscopes that provide us access to high resolution data in live samples. And as with many things in life, you have a lot of trade-offs when you develop new technologies. And in microscopy, we summarize these trade-offs often in what's called a pyramid or a triangle of frustration, because there's no free gains over here. And our goals are to, of course, go faster so we can look at these molecular scale kinetics, be at high resolution so that we can go past this kind of limits on the spatial scales that we have to access molecular interactions. And then, of course, go deeper into samples so we can look at intact embryos and tissues, and then look at many different colors at the same time so we can observe molecular interactions between different proteins. When what we need in every microscope is good contrast, because if we don't have good contrast, then we cannot even visualize our signal above the background noise. To get good contrast, you can increase, for example, your exposure time or your laser power that you use to excite. But in the process, then you reduce the duration of your imaging because of the photo bleaching that we discussed. Now, if you want to go really, really fast, you have to have short exposure times or high laser powers and high laser power. So then you bleach your sample very quickly, again, limiting the duration. Same thing with spatial resolution. If you try to shrink the volume that you're trying to get signal out of, your contrast will drop because there's less fluorescent molecules in that volume. And as a result, you might want to increase your laser power, again, limiting duration or the contrast. So in my lab, what we do is we focus on this one vertex of this pyramid called the contrast vertex. And by optimizing along this axis, we have to make less trade-offs in spatial resolution and temporal resolution and duration. So the way we do this is by using a type of microscopy that's very different than those of you who do microscopy might be used to. In a typical fluorescence microscope, like I showed before, you're sending light through the sample, and it's exciting all the fluorescent molecules within your sample. Now, if you use a high numerical aperture, a high resolution objective, you will get a thin slice of these molecules in focus where you can make an image. So all of these things that are out of your focus are still absorbing light and emitting photons, but they're not actually contributing to your signal. Instead, they're contributing to your background. So it reduces your contrast. So instead of illuminating the sample this way, the basic idea is that we illuminate the sample with a thin sheet of light. And we optimize the thickness of the sheet of light so it matches exactly the plane that we're imaging, so that no light gets wasted in this process. Now, in practice, to pull this off is actually quite a bit of effort. We use a type of light sheet microscopy called lattice light sheet microscopy. And the video you're watching is of my lab's construction of our state-of-the-art light sheet system built based on designs from Eric Betzig's lab at the Janelia Research Campus. This microscope can do quite a few different things. And as you can see, it's quite a complicated machine. But at its heart are these two orthogonally positioned objectives. One microscope objective is used for excitation, using this exquisitely thin sheet of light. And then the second objective is used for detecting the fluorescence from the molecules that are excited by this thin sheet of light. And this method of selective plane illumination and detection allows us to have really, really nice contrast deep inside of samples because we're not contributing to the background. And this in turn allows us to go either faster or for longer periods of time because we have limited photo bleaching. And just to give you an idea, this is a comparison of a spinning disk confocal microscopy, which is considered the gold standard for live cell fluorescence imaging, compared to lattice light sheet microscopy. And you can appreciate how the signal fades very quickly in the spinning disk. while at the same sort of imaging settings, and that is light sheet microscopy, you can maintain the signal for much longer. Now, we solved the contrast problem, but this does not allow us to actually access the spatial scales that we're interested in. Remember, we want to understand how these transcription factors are finding the targets, and this is just 100 nanometers square. So the question is, why can't we just keep magnifying our images until we can see these molecules? And we can't just keep putting microscopes on tops of microscopes because of something that's called the diffraction limited resolution of light. And this is actually literally written in stone in a monument to Ernst Abbe, who derived this equation of resolution in light microscopy, which is that the smallest distance between two features in your image that you can resolve is about half of the wavelength of the light that you're using to image them. So if you're using green light at, say, 500 nanometers, you would only be able to tell things apart from one another if they're about 250 nanometers apart. So this is just frustratingly a few hundred nanometers beyond the scales that we're interested in accessing, that below that 100 nanometer scale. So this problem was solved in a variety of ways in the last couple of decades. The method that we use is called a single molecule localization microscopy. And Eric Betzig, who I mentioned earlier with the light sheet design, also won a Nobel Prize for this, developing this technology back in 2014, called PALM, photoactivated localization microscopy. And the basic idea over here is, instead of trying to tell two things apart that are less than 250 nanometers apart in one single frame, you make your molecules or your fluorescent molecules blink in a way that you can localize one of them with nanometer precision by pointing to the center of it. And then it shuts off. And then you activate the next molecule next to it, and then you localize that. And this way, by kind of going through every single molecule in your image, you can reconstruct a high resolution image. And this is illustrated nicely in this movie of the light show on the Eiffel Tower, where the lights are blinking. And by pointing to the center of each of the lights over time, you can reconstruct the shape of the entire tower. So this is very good if your sample isn't moving, so you can make a high resolution image. But what we do is we put a twist on this technology for live imaging, in that we use the same principle of photoactivation. So these are molecules that you can turn on very stochastically, and we turn them on at such a low level that the probability of more than one molecule being on next to another molecule within your resolution limit is basically zero in every frame of your movie. And then while the molecule is on, we can image it and follow it around because we have that very nice contrast. And we can point to its center with 20 nanometer precision. And this is what it looks like when we combine this photoactivation technique with light sheet microscopy. What you're seeing over here in this movie is in green are single molecules of a transcription factor protein inside of an embryo. You can see the edge of the embryo over here. And in red, in the middle over here, what you're seeing is chromatin, DNA, which is visualized using the histone H2B. And you can see these molecules sort of explore the environment. Sometimes they stick for a little bit longer. And then we can do similar experiments in all sorts of embryos. Here's an example of a mouse embryo where we're looking at a protein called CTCF inside of a very early stage mouse blastocyst. So these type of movies, to me, are still mind-blowing, that we can look at single molecules inside of a living animal embryo. But I think what's really much more interesting is what we can do with these type of data. So we have algorithms to now localize the molecules in each frame of the movie and then connect them into trajectories. And we can see molecules that don't move very much and molecules that move more. And then by applying biophysical analyses, which I'll discuss very briefly as I go through our data, we can begin to ask questions about what type of mechanisms allow these proteins to efficiently explore the nuclear environment, how long they stay at their targets when they find them, and how long does it take to actually find their targets in the first place, and so on. So our interest is applying these technologies in the context of a process called zygotic genome activation. So following the fertilization of an embryo, the zygotic genome, which is the genome of the fertilized embryo, is transcriptionally silent. And all of its life is maintained by proteins and other products provided by the mother, loaded into the mother as the egg is initially made. And this process is conserved through all animals, although it happens at different timescales. And basically, as you proceed through early development, the maternally loaded products slowly activate the zygotic genome, and it begins to take over in this process of zygotic genome activation and prepares the embryo for independent life. And today I'll be talking about this process in the context of the fruit fly embryo, Drosophila melanogaster, which is the powerhouse in developmental biology and genetics because of our ability to manipulate it genetically and all the tools that exist to understand these basic fundamental molecular scale processes during development. So the embryo itself is about just 0.5 millimeter in size. And in these early stages, within the embryo, you have a nuclei in its syncytium, so there's no cell membranes. And the nuclei undergo a series of very rapid nuclear cleavage cycles. So every 10 minutes on average, they divide and make more nuclei. And basically, although all of these cells look the same, at the transcriptional level, they're all encoding different genes at this point and beginning to pattern the embryo. And this happens in this very short time frame, in just 3 hours, going from the fertilization of the egg to differentiation. So it's a really high throughput, powerful system to understand these processes. So this process is kicked off by these maternally deposited factors. And some of these are distributed in concentration gradients called morphogens. And these concentration gradients turn on genes that encode for other transcription factors. And there's this sort of hierarchical network of activation and repression that progressively patterns the embryo into more and more defined domains of gene expression, which after gastrulation, which is the initial folding of the different tissues in the embryo, they go on to form the different body segments of the larval stage and eventually the adult fly. Now, what we want to do is understand these interactions at the molecular scale and see how the genome is actually interpreting things like a concentration of a factor. Now, we'll focus initially on the earliest known morphogen, which is a protein called bicoid, which is actually the first known morphogen in the world, and is distributed in a concentration gradient going from tens of thousands of molecules per nucleus in the anterior or the future head of the embryo, down to tens to hundreds of molecules per nucleus in the posterior or the future tail of the embryo. And what people have sort of thought for decades on how the cells now read out this concentration to make decisions is that within the genome, you have elements that are responsive to all concentrations, elements that are only responsive to the that are more sensitive, that are less sensitive are on at the anterior. The ones that are more sensitive begin to shut off as the concentration drops. And these sort of models predict a variation in how long each bicoid molecule would bind to these genes to turn them on and off. So what I initially set out to do while I was a postdoc was to measure these off rates, so how long a molecule sticks on a gene, so its residence time, and then the on rate, how frequently it sticks to the same gene. So to do this, we acquire single molecule data. So here you're seeing molecules that are immobile, and then we track their lengths of each of these detections, and we build what's called a survival probability curve, which tells you what's the probability of finding a trajectory or an interaction of a certain length. We fit this to various biophysical models, here a two-state model of specific and non-specific interactions, and then we correct for effects like photobleaching and defocalization probability, and from that we can extract a specific residence time of bicoid of just 1 to 2 seconds. So the longest interactions that we observe are on the order of 2 seconds or so. So these molecules are popping off of chromatin very, very quickly. Now, if you were to imagine as an engineer, if you're to design a system that is sensitive to concentrations, this would make a lot of sense. You want molecules to pop off very quickly so that when you're at high concentrations, another molecule will quickly reoccupy that site, leading to a high time average occupancy of the site that you're interested in turning on. At low concentrations, if a molecule pops on and off very quickly, it would take a long time for another one to find the same site, so that then you would have a low time average occupancy. So then your occupancy depends solely on concentration, and that would be fantastic. But our data told us that this is unlikely the way this is working, because when we use another technique to measure how much of the molecules or how much the fraction of molecules is bound, what we do is we measure the distance between individual localizations in our trajectories. We plot these displacements into a distribution, and we model this distribution in a two-state diffusion model, where we can extract the fractions of molecules in either a slow population, which is on these low displacement ends, or in a fast-moving population, which is these larger displacements. And from this type of analysis, what we find is that the diminished population of bicoid at the posterior of the embryo, where it's low concentration, is still bound to DNA. And then we wondered if this binding is to specific sites, or is this just kind of non-specific interactions with the DNA. So we performed an experiment called ChIP-seq, chromatin immunoprecipitation and sequencing, where you basically pull down the pieces of the DNA where your protein of interest is bound to, you sequence them, and you map it back to your genome to figure out where proteins are bound. And we found that in the posterior of the embryo, at these low concentrations, this binding is to known target sites. And from a biophysical perspective, this is a huge challenge, because you have low concentrations, very high off rates. How do you solve this problem? What we found was that the way life has solved this problem is by concentrating proteins to specific regions within the nucleus. So these are kind of 3D maps of bicoid detections of this molecule within a nucleus. In the anterior high concentrations, you see that these detections are clustered, but they're all over the nucleus. In the posterior nuclei, you see a fewer clusters, but their densities begin to match what you see in the anterior. So we hypothesize that this clustering is now concentrating molecules around the genes that need to be turned on in the posterior of the embryo to drive the occupancy that we see with single molecule or with our genomics techniques. Now, we became very interested in understanding how this clustering is actually being achieved at the molecular scale. What is telling these molecules to go to where they need to be? So our first hint came when we deleted another transcription factor called ZELDA. And when we delete this transcription factor, these clusters disappeared. Now, ZELDA is a very interesting protein. It's an acronym for zinc finger early Drosophila activator, because it has 4 zinc fingers, which comprises DNA binding domain, and the rest of the protein is more or less unstructured. And what ZELDA is it's sort of a master regulator in the early embryo. It's not a morphogen, so it's uniformly distributed in the embryo, but it dictates the binding of many, many transcription factors, and in doing so regulates these domains of expression that pattern the embryo. So we thought initially that ZELDA is now sitting at these gene targets where bicoid needs to be, and then through these kind of unstructured domains, reining in bicoid to these sites to cause the clustering that we observe. So when we measured the residence time of ZELDA, we were really surprised to find that it also sits on DNA just for 5 seconds or so, just a little bit longer than bicoid. So it does not sit there stably and recruit bicoid. So instead of doing single molecule, we asked, okay, how does ZELDA look in the nucleus overall? So now instead of doing single molecule, we do very high-speed 3-dimensional imaging using the lattice light sheet microscope. And what you're seeing over here, this is sort of the edge of the embryo. And within the embryo, each of these balls is a nucleus. And in gray, what you're seeing is the spatial distribution of ZELDA. And now this nuclei are dividing. And as you come back into the next cell cycle, you'll see that ZELDA rapidly distributes into these local high concentration regions that we refer to as hubs. Now, these regions are very transient, but you can appreciate how rapidly the concentration within the nucleus in a specific part of the nucleus varies in space and time. So now, if you're a gene along that 2-meter polymer sitting in here, the concentration of your activating protein that you see can vary very quickly in space and time. And that's what we now want to understand, is how this change in concentration is actually connected to transcriptional activity. So to do this, we adopt another technology. And this technology was developed by Rob Singer's lab at the Einstein Institute in New York City, which is called the MS2 system. It's a really cool piece of technology in which we take a sequence from a bacteriophage, which is a virus that infects bacteria, and we insert it into our gene of interest using genome editing. When this viral sequence is transcribed, it makes a very specific stem loop structure. Now, this stem loop structure is recognized by the coat protein from the same virus with very high affinity. And what we do is we fuse this coat protein with the fluorescent protein molecule. So then when the gene is transcribed, we get a signal for where the transcription is happening. And if we have multiple repeats of the sequence, we also have a readout of the level of transcriptional activity. So now combining this with our high resolution imaging, we can see how these transcription factor hubs are related to transcriptional activity. And so these movies look like this, where in white, now you're again seeing the spatial distribution of Zelda. And in orange, you're actually seeing live transcriptional activity at one of Zelda and Bicoid's target genes called Hunchback. So again, here we were surprised that these hubs of proteins aren't stably interacting with these transcription sites. But we devised a method to see if the interactions are different at that site versus other sites in the nucleus. And what we do is we use the transcription site as a sort of anchor, and then we see how the protein signal around it is varying versus a random site in the nucleus. And this is what that analysis looks like. And what we see as we start to average the signal is that you end up with an enrichment of the protein at the transcription site versus random sites in the nucleus. And what this tells us at the end is that these Zelda-dependent hubs of Bicoid are preferentially interacting with their target sites, leading to an enrichment of Bicoid at that locus compared to random sites. Now, to those in the field, this is not surprising that a transcription factor is going to the site that we know it's regulating. But I think what's really surprising is what we learned about how it gets there. It gets incorporated into these hubs, which then transiently interact with their target site and drive occupancy. So we have basically sort of kind of kicked our problem down the road, because we started by asking how are transcription factors targeted to their sites in this complex nuclear environment. Now we said that they're concentrated into hubs, but the question now is, how are these hub properties being regulated to target these hubs to different genes? Are there multiple genes there? Are they assembling at the genes? Are they assembling elsewhere and then being targeted? So to start addressing this question in my lab, we first asked, how are these hub interactions being, are they specific? And if so, how are they tuned to drive different levels of occupation at different gene targets? So my graduate student, S, used another morphogen system within the embryo, which is from a protein called dorsal, which instead of the head to tail or anterior posterior axis of the embryo, you look at the dorsal ventral or the belly to back axis of the embryo. Now, dorsal is really cool because at each nuclear division, it gets pumped in very rapidly into the nuclei. And as it gets pumped into the nucleus, we begin to see that it forms these distinct high concentration regions. There's also puncta outside of the nuclei, but we can ignore those for now. That's not relevant to this story. But within the nucleus, you see they very quickly form as the concentration is increasing. So S asked, are these hubs localizing to their specific target sites? So first, they characterized how the hub formation is occurring. And they found that the hubs begin to form very soon after the protein enters the nucleus. And then by using this MS2 system that we just discussed at different target genes, S observed that these hub properties are different at different dorsal targets. And in this case, we use hunchback as a negative control because dorsal is not supposed to regulate its activity. Now, one of the most interesting out of this set of genes is a gene called snail. And if we zoom into this movie, what you might notice is that if in pink, you're seeing the gene being transcribed, and in green, you're seeing dorsal, you might notice that one of these dorsal hubs is sort of following the transcription site around. So you can imagine the DNA polymer in here. That's the gene that's being read out. And around that gene, we have a hub of dorsal that is driving presumably transcriptional activity. So by applying the same analysis on using the transcription site as an anchor point, what S discovered is that these hubs lead to different levels of occupation at these different gene targets. So the enrichment is tunable and is specific such that you don't see any enrichment at the negative control here, hunchback. And digging into this data further, what S found is that these hub properties are like lifetime of the hub, or the persistence of the interaction is also tuned at different target genes. So longer interactions lead to longer enrichments. And this corresponds very nicely to what you can observe with techniques like ChIP-seq. So we think we have unveiled a mechanism by which explains how you get different occupancy at different target genes, which is by tuning the enrichment and the lifetime of these transcription factor hubs at their target sites, which then leads to a higher level of opportunities for the factor to bind, leading to these high levels of occupancy. So I get a question often that how widespread could this mechanism be? Is this something unique to Drosophila embryos? And to sort of address this, we looked at across the literature at hundreds of papers. And what we did was compile the residence times reported for different proteins. And what we found is that throughout life, going from worms to humans, through many, many different types of transcription factors, the residence times of these proteins on chromatin does not vary very significantly. At the same time, for those in the field who are probably thinking, there's this explosion of reports in the literature about condensates and other structures that concentrate proteins. And anything that we look at in the fly nucleus actually shows that it's not uniformly distributed. But the substructures that are being formed have very different levels of stability and enrichments, pointing to likely different mechanisms of formation. And this goes from hours of stability down to seconds. So this is what life is modulating, not the stability of specific protein interactions, but rather the enrichment and stabilities of these higher order structures. And in collaboration with the cancer biology group on campus, we've also now shown that when you make a mutation to a regulatory protein, which is found in Wilms tumor, a pediatric kidney tumor, as well as in forms of leukemia, it takes the wild type distribution of this protein with the nuclei from being relatively diffuse to being now concentrated in the mutant very, very stably at the genes that are upregulated in these different types of cancer. So these mechanisms of tuning the hub properties and stabilities, they're very finely tuned for their functional purpose. Now, at the end, what all of this points us to is this idea that it's not the stability of interactions, it's the frequency of interactions that can lead to differential occupancy and different levels of gene activity. And together, we can reach this kind of idea that the biophysical properties of these different environments are tightly controlled and are very tightly linked to their functional outcome. Now, in the lab, we're beginning to address what is driving this hub formation at different target sites. And we returned back to Zelda to answer this question. And what we did first is we posited, because of a lot of reports in the literature, that the intrinsically disordered regions, these are the unstructured parts of the protein, actually play a big role in the targeting. So we mutate the DNA binding domain and see what the rest of the protein does. And what we found is that, as expected, when you mutate the DNA binding domain, first, the stability of the protein on DNA goes down. So if you look at the residence time, it goes from that 5 seconds just down to 1 to 2 seconds. And this protein kills the embryo. but it still provides us some insight into how the rest of the protein might be working. So when we look at hub formation through the different stages in development and compare the wild type protein to this mutant protein where we got rid of the DNA binding domain in effect, what we see is that the hubs can still form. So the hubs formation is not necessarily dependent on the stable protein DNA interaction as we suspected. Then we look genome-wide using next generation sequencing, so RNA sequencing, and a technique called cut and run to see where the protein is binding. What we find is that when we mutate the DNA binding domain, the gene expression patterns within the embryo change. So some genes are downregulated and some genes are upregulated. And the genes that are upregulated actually are now occupied by this mutant protein when they were not occupied by the wild type protein. So perturbing the DNA binding domain kind of redistributed the protein within the nuclear environment and redistributed to genes that are now transcriptionally active. So the model that we're getting at on how transcription factors find and occupy their targets is that there is this really fine kinetic balance within the nucleus that is dependent on a hierarchy of very transient, preferential, low affinity interactions. So on the genome, you have proteins popping on and off a site very quickly. But if you have a long strip of such sites, you can drive a high local concentration at that site. But if you perturb the sort of center pin of this interaction, which is this protein Zelda, by mutating its DNA binding domain, then this interaction at a probability at this gene locus gets weakened. And all the proteins can then go to the next strongest site, which may be occupied by weaker DNA protein interaction, but now takes over from where Zelda was initially driving high occupancy. Now, we think that this data is really forcing us to rethink how transcription factors actually work, because for decades, we've assumed that they're structurally and functionally modular, in that you have a DNA binding domain that tells the protein where to go, and then you sort of have this disordered activation domain that is responsible for recruiting the rest of the protein machinery. But I think the data that we have shown tells us that this activation domain and DNA binding domain work together to drive the specificity of where these proteins go, and now represent to us a new target that we can sort of manipulate or perturb for therapeutic or engineering purposes. So at the end, we have a new vision of how the nucleus is organized, which is through this sort of very, very low affinity interactions. And this is very exciting because this enables us to now tune these local proteomic environments and these reaction probabilities to tune transcription, and also provides us on basic insight on how over the course of time you can have very different phenotypes in related species without altering much of the genome or much of the proteome, but maybe by having subtle mutations in these higher order interactions in the disordered domains of proteins. So with that, I'd like to leave some time for questions. So I want to thank my wonderful team in my lab. We're a very interdisciplinary group. I have only talked about one of our projects, but we're doing a lot of technology development as well, and looking at various aspects of nuclear organization. And our funding sources, which is the National Institute of Health, the Howard Hughes Medical Institute, the National Science Foundation, and the Margaret Q. Landenberger Research Foundation. And again, thank you all for your time and attention, and for this opportunity to share my work with you all. So happy to take any questions.
Thank you very much, Dr. Mustafa, for your wonderful presentation. We have time for a few questions. So if anybody has any questions, you may type it in the chat, please. So, sir, we can see very lot of praises for very informative and interesting session. But if you have any questions, if there are any questions from the participants, we're ready to take them.
Yeah, happy to talk about any aspect, the microscopy, the technology, the biological insights. Don't be afraid. You can also feel free to email me. You can look at our website and find my email address if you think of anything later on.
Yeah, usually, so when somebody has no questions, it means the presentation was very
Yes,
self-explanatory.
everything was very clear.
Yeah, and they understood everything. However, we would appreciate if they have any questions or any feedback. But anyways, Dr. Mustafa, thank you very much for joining us. I understand that it's very early in the morning in the US, probably like 7 a.m.,
Not a
right?
problem. Not a problem.
Thank you very much, sir. So yeah, I suppose nobody has questions here. All right. Thank you very much, Dr. Mustafa, for sparing your very precious time, very informative session. And thank you very much to our audience that have joined us from several OIC countries. We are very glad to see you all here. Thank you very much for your time and hope to see you in future important events. Okay, and just as we're about to leave, we have one question from Mr. Irfan. He's saying, have you tried another type embryo?
Yes, we're looking at many different types of samples at this point. We're doing a lot of work in mouse embryos now. We've worked in C. elegans as well. We're very excited about what we're doing with the mammalian embryos because, of course, it's closer to what happens in other vertebrates like humans and has sort of, we can ask different questions there than we can do in the Drosophila embryo. We're also looking at a lot of different cancer models, usually in sort of blood cells. So we're looking at kind of non-inherent cells using the light system as well.
Right. All right. I think if anybody has any further questions, you are free to get in touch with us or get in touch with Dr. Mustafa directly. All right. Thank you very much, sir. Thank you.
Thank you very
Thank
much.
you very
Take
much
care.
for your time. Goodbye. Good. Take care.